Hello and welcome to our podcast Pager. I'm George Milner.
And I'm Stan Dale.
Tune in to hear us talk with some amazing people about the subjects changing medicine today.
From stem cells to mapping the brain and medical ethics, we hope to give you a glimpse of the bright future ahead through discussions with some of the people making it happen. Hi everyone, welcome back to another episode of the podcast. This week I was lucky enough to speak with Dr. Stephan Kissler about how genetic tools are being used to deepen our understanding of type 1 diabetes. Now, Stephan is a principal investigator at the Joslin Centre for Diabetes here in Boston. He graduated from the University of Bristol with his Ph.D. in Immunology before moving to Boston to do his post-doctoral work at the Dana-Farber Cancer Institute as well as at MIT. He was appointed to a group leadership at the Virchow Center in Germany in 2007 before coming back to Boston in 2012 to start his own research group at the Joslin. Which, by the way, is a really interesting place. It's a really world-class research institution and is somewhat unique here in Boston in that it's solely focused on diabetes. And we're able to talk a bit about its now long history in the podcast. I've always thought that type 1 diabetes is a particularly interesting disease in that, whilst it's known that type 1 is caused by an autoimmune destruction of the insulin-producing cells in the pancreas, the beta cells, when you scratch beneath that answer, you realise that there is still lots to be uncovered about many aspects of the disease pathogenesis, and also about which potential treatments and therapies might be useful for slowing its progression or for even stopping its progression. Now, Stephan first talks to us about his work looking at genes identified in association studies back in the 2000s. Now, these were new genes that no one had necessarily associated with type 1 diabetes before. And he used genetically modified NOD mice, which are the primary animal model for type 1 diabetes, to look at these genes. We then go on to talk about Stephan's more recent work, in which he essentially switched things around, and rather than looking at the effects of a single gene in the whole organism, he instead looked at changing every single gene in the beta cell, the cell which is destroyed in the disease, using a Crispr screen. Later in the episode, we also managed to get into a discussion about stem cell-derived beta cells and the pros and cons of a personalised approach for patients, which would use their own cells to treat the disease, as opposed to a one-size-fits-all, off-the-shelf cell therapy. We're also even able to get into how Stephan developed different skills throughout his career in order to differentiate himself as a scientist in what is a very competitive field. So the hour is really packed with all sorts of topics. Anyway, without further ado, we hope you enjoy our episode with Dr. Stephan Kissler. And we're live. No, I'm just joking, we're not live. Stephan,
with an audience of 10,000.
Stephan, thank you for coming on the podcast.
Absolutely, you're very welcome.
Today, I suppose we're going to talk about type 1 diabetes. That would make the most sense. You lead a research group at the Joslin, which actually we were just speaking about the Joslin off the air. It might be nice to talk a bit about it as a centre before we start. I think that could be fun. It's like a very impressive and unique place. And your research is focused around sort of pathogenesis of type 1 diabetes, an autoimmune disease affecting the pancreatic beta cells.
Right.
And I suppose your work touches on a number of aspects related to T1D. And one of the things I wanted to ask you about was sort of maybe some of the more tangential projects your lab's working on, as well as Pathogenesis. But perhaps just to start, you could talk to us about what your research group does and also how you ended up sort of leading a group at the Joslin.
Yeah, so maybe let me just very briefly tell you about the Joslin, because I think it's a really fascinating place historically. So it's named after a doctor called Elliot Proctor Joslin, who started he was a medical student in Boston at Harvard, and then started a diabetes practice focused solely on diabetes, and that was before the discovery of insulin, which means that if you had diabetes, particularly type 1 diabetes, it was more or less a death sentence, because you can't live without insulin for any length of time. Usually people would pass away within a couple of years if they were that lucky, unless you were on a very strict starvation diet. And so out of this practice that was on Bay State Road, which is near Boston University, this practice grew. It was just a town home where Dr. Joslin lived and practiced medicine, and it grew so much that eventually it became too big to be hosted in this one building, and they actually built a new dedicated clinic that is now the Joslin Diabetes Center, and that was built in the 50s. and then with some additions in the 90s, and we're now in the middle of the Harvard Medical School medical campus where all the hospitals are located. So it's just a really interesting history, because it started with one doctor, right, who was fascinated. He was a leading figure in the diabetes research or treatment world in the early 20th century, and it grew into this world-renowned center for the treatment and research of diabetes. Yeah, I mean, the building itself, actually, if you've seen the buildings around campus, our building is probably the least impressive of all. It's very old.
I was going to say.
And it's small. So we're surrounded by all these big hospitals, Beacon Deaconess Medical Center, the Dana-Farber Cancer Institute, that had these huge brand new buildings across the street, the Brigham Women's Hospital, Children's Hospital. They all have much bigger facilities than we do. But we're very specialized, so we're very focused. So everyone inside this building is working on some aspect related to diabetes. So we have a diabetes clinic that treats patients, and then we have researchers, and all of research is related to diabetes and its complications.
I think the advantage of the Joslin is that if it's a sunny day and you're in Longwood, it's about the only building you can look at without being blinded.
Our laboratories are, they're kind of a classic design with wooden benches still, so pretty old school. But we feel like we don't have to have the newest facilities to do good research. It's all about what you do and not your setting itself. But we're very happy there. So, and one of the advantages of being at Joslin is that it's a relatively small community of scientists. So within Joslin, you get to know everyone. Unlike in some of the big hospitals where you have thousands of researchers, and it feels a little bit more anonymous at times. So we have a very tight-knit community of researchers, and we interact a lot, collaborate with our labs a lot within the building.
It must have been an exciting place to sort of just be around pre-pandemic, in terms of talking to people who are working on similar things to what your lab's working on, and bouncing ideas off each other. Going back to Dr. Joslin, sort of, who you spelled out, sort of ended up founding this whole institution. If this was pre-insulin, was his treatment more focused around sort of diets which prevented spikes in insulin? So
right, absolutely. So I think if you go back, so diabetes is a disease that has been known for thousands of years. There's references to diabetes in the old Egyptian cultures and beyond. So it's nothing new. But as I said, if you had diabetes back then, you were pretty much condemned to death. And even just 200-300 years ago, there are records of special diets given to people with diabetes. The way they recognized that someone had diabetes back then was by tasting the urine, which tastes sweet, or you can ferment it with yeast. Actually, I have these old case reports from the 1700s of doctors describing the cases of diabetes in their patients, saying how they fermented their urine with yeast, and that was a sign that there was sugar in the urine, and so that that was a sign of diabetes. Or you could evaporate it, and then you end up with sugary residues that looked like molasses. And so that's how they measured how much sugar was in your urine back then. And so based on this, I mean, and I think a lot of it was just trial and error diets. People were recommended diets that were very poor in carbohydrates, meat based, for example. And so that's all you could do, is to tell people to eat less food that was likely to spike their blood glucose and their urine glucose back in the day. But eventually, this is a kind of diet that wasn't sustainable, so a lot of people would basically starve to death. So that and that was until the discovery of insulin in 1921, which of course completely changed everything. With then people being able to take insulin every day and lead a relatively normal lifestyle, if you like.
And we can talk probably a bit more about perhaps some of the changes that have occurred since the discovery of insulin, in terms of recombinant insulin and all of that sort of stuff. But perhaps you could just tell us how your career path has led to you being at the Joslin. Sort of, where did you start your scientific career? How did you even get interested in sort of science and biology?
I have a background in science from my family. So my family has a lot of scientists in it. My father is a retired physicist. My grandfather was a physicist. I was surrounded by a lot of science when I was a kid. But nothing. I wasn't the kind of kid who was. I wasn't always just focused on science myself. But it was just omnipresent. So my father worked at the CERN, the European lab for particle physics in Geneva. So that's where I grew up. So, for me, it was kind of normal to have science talk around. But when it came to decide what to do in college after high school, I really didn't have a very specific ideas until the last few months before I had to commit. Soon, close to graduating high school, and I decided that biology had always interested me, and so I decided to start studying biology, and with a sense of really just open mind as to what topics in biology I would study, plants and birds and animals and all kinds. So I had a very naive idea, if you like, starting with studying zoology, botany, animal behavior, paleontology. So it was a mix. So I started that in Zurich in Switzerland for a couple of years. Before then, starting to focus a little bit more on kind of, if you like, modern biology, which biochemistry, molecular biology, rather than the chasing butterflies around the Swiss Alps, which we did it too. But and then I moved to Bristol in England for a couple of years, where I actually graduated with a degree in biochemistry. And during one summer, I had the opportunity to do a summer internship in a lab that was studying immunology and rheumatoid arthritis, so the autoimmune side of immunology. So rheumatoid arthritis is an autoimmune disease, just like type 1 diabetes. And so that's the first time I was kind of, I encountered these really fascinating concepts of, you have an immune system, it's there to protect you, but sometimes it goes haywire and it starts attacking your own body. And I thought that was a fascinating subject. And the immune system is obviously very complex to understand. And it just posed an interesting challenge, if you like, scientifically. So that kind of spawned my curiosity in this. So I ended up doing a PhD then in immunology, studying autoimmune disease and multiple sclerosis in particular, using mouse models.
Where were you for that work?
And I did that actually
in Bristol.
Yeah, that was in Bristol as well. So actually, I left for after I graduated my degree in Bristol. I went to Germany in Heidelberg for a year to work on an HIV project as a research assistant, and then I moved back to Bristol to do a PhD in immunology with David Wraith, who's a leading figure in the multiple sclerosis world. So there, I got really interested in T-cell biology. And the whole kind of autoimmunity that underlies multiple sclerosis and many other diseases. And after that, then I did what a lot of young PhD students do in Europe. They go to the US to do a postdoc because that's the land of opportunities and big name scientists. So then I came to Boston, and I spent a couple of years at the Dana-Farber Cancer Institute working in a lab that was very focused on T-cell biology. And then I went over to MIT for 4 years, where I did a postdoc learning about genetic engineering of mice. I actually started making mice with engineered genomes where you could turn off genes at will. And so that's what I then pursued for the rest of, since then I've been making mice with targeted mutations so we can study specifically genes involved in autoimmune disease. So we've done that for many years now.
In a sense, you can see the seeds of sort of some of the really interesting papers your lab has done in the last few years in your postdoctoral work, I suppose, because sort of, for example, the models looking at beta cell proliferation and how that can actually protect against type 1 diabetes is very dependent on these conditional knockout mouse models that it sounds like you were honing those skills whilst you were doing your second postdoc.
Well, I think that in my second postdoc at MIT, I really expanded my skill set, if you like, because I was using a lot of classical immunology methods, cellular immunology, that just consist in taking cells out, looking at what they do, and then put them back in and looking at what they do in the mouse. And I actually gained some skills in engineering mice, so I didn't rely on actually models that other people made, and it gives you a certain independence of picking any topic, any gene, and saying, I'm going to look at that gene, and I will make my own tools to study that gene, and so you can make your own mouse models. So you don't rely on what has already been done.
And now we're at a point where the genetic models are like much broader in terms of fishing for genes that might be interesting. Sort of this brings to mind another paper of yours, which I'm sure we'll talk a bit about, where you can use CRISPR screens to essentially look for any gene in the genome which might have an effect on something you're interested in. So, for example, beta cell resistance to stress.
Yeah. And so these are two very different approaches. You can take a very focused approach and study a single gene and really dig in and try to understand what it does. Or you often, if you don't know what to study in the first place, you'll just screen, for want of a better idea, if you like, to see if anything out there in the genome is interesting for your particular question, which we did for this project that you mentioned, and we can happy to talk about it in more detail. But I always thought it was, this was a kind of a turning point in my career, if you like, to learn the skills to make my own genetically modified mice, because then you can really start exploring questions that no one's addressed. You can start interrogating genes that no one's ever looked at. The function is unknown, and you can really go into uncharted territory, if you like, because if you only rely on models as others have made, for the most part, the kind of the low-hanging fruits in these models have already been published. So people will make their knockout mouse and they'll study a gene and they'll publish the best results, and then you too can use that mouse for your own studies, but you're never going to discover anything that's fundamentally new. Whereas if you make your own targeted models, then you can really go after genes that no one's ever looked at. So it's kind of, for me, it was an opportunity to start looking at genes that no one else necessarily had studied before, particularly in the context of diabetes. And the way I got into type 1 diabetes actually is simply by acquiring these skills of making mice, and we start collaborating with a laboratory that's actually in Cambridge, UK, or was at the time, Professor Linda Wicker, who was an expert in type 1 diabetes, a leading figure in the mouse models of type 1 diabetes at the time. And I collaborated with her, and that kind of got me, sucked me into the field a little bit. So I already had a broad interest in autoimmune disease, and by collaborating with this leading figure in type 1, it kind of gave me motivation to actually get to know the field a little better, understand the diabetes models better, understand the disease better, and the questions that might need answering to cure type 1 diabetes. And I think all the different autoimmune diseases have a lot in common. Both genetically, there are a lot of risk genes for autoimmune disease that are actually shared between multiple sclerosis, rheumatoid arthritis, type 1. So in my mind, studying one autoimmune disease was going to inform you about many other diseases. If you figure out how one works, you may be able to apply some of these findings to other diseases. So that's why, in my mind, it didn't really matter if I studied multiple sclerosis, type 1 diabetes, rheumatoid arthritis, because they had a lot in common. And the second reason why I then got into type 1 diabetes is really because I saw an opportunity to apply my new skills of engineering mice, applied specifically to a model of type 1 diabetes that's notoriously difficult to manipulate genetically. So we use a particular strain of mice called the non-obese diabetic mouse that's been around for about 40 years now, and it's been the prime go-to model to study type 1 diabetes in animals. And it's always been very difficult to manipulate genetically. So it wasn't as easy to make knockout mice and knock-in mice in that model compared to other models that people use for other diseases. And so my skills were really suited to manipulate that mouse model. And so I saw an opportunity, if you like, a niche where I could maybe make a mark and be competitive. Because as a scientist, you always want to do good science, but at the same time, you also in your career, as you grow, especially in the early stage of your career, you have to define where you're going to be competitive and where you can make your mark in the scientific world, because there's a lot of scientists out there. Everyone's trying to do good work. Everyone's trying to publish good work, and it's not always easy to define yourself and be a little, have your unique expertise recognized by others. So you have to.
I've always thought that a PhD is sort of. It's one of the most prestigious degrees you can get, which doesn't confer any job security at all. You're only you really have to keep selling yourself as a scientist. So it sounds like you arrived in type 1, perhaps as a combination of, collaborating with someone who was somewhat sort of a leader in the field, and maybe I find that probably quite inspiring, I suppose, coupled with seeing really an opportunity where actually your skills modifying genetically modifying mice is really useful, considering so much work is done in this particular model you mentioned.
Right, and I think particularly the timing was very right for this. Just at the time when I was acquiring these skills of manipulating genes in a model for type 1 diabetes, the first genome-wide association studies had been done for type 1 diabetes. And what this is the first studies where you could compare thousands of people with type 1 diabetes and 1,000 people who didn't have the disease and compare their genomes and try and find those genes or genomic regions that associate with the risk of disease. And so the first time this was done was in the early 2000s, about 2005, 2007. First papers started being published on this large survey of what genes actually increase your risk of having type 1 diabetes. And all of a sudden we had this long list of genes, dozens of genes, that were shown to increase the risk of diabetes in people. And yet a majority of these genes had never really been studied in the model. And some of these genes, no one even knew the function of the gene. Some didn't even have a name. And so this was a great opportunity to then apply these skills of manipulating genes in a mouse model, because you could say, I have this long list of genes, we know they're involved in disease, but we don't know why. And now is a good time to start studying them. And so I almost I decided that I would pick a number of genes that kind of looked interesting to me, maybe because they hadn't been studied as much as others, maybe because they were also associated with additional diseases, not just type 1, but genes that seem to have an effect on both type 1 diabetes, multiple sclerosis, rheumatoid arthritis, Crohn's disease.
Presumably GWAS studies were being done in all of these other diseases as well at sort of similar times.
Right. So the GWAS studies all started coming out, being published within a few years of each other for all these diseases, and you saw a lot of overlap in autoimmune disease. And so I started studying these genes, and I picked a few, and then we made mouse models for several of these genes, and then studied the function of the genes in disease. And so that was kind of a good starting point, I think. So just at the time when I was starting my own independent laboratory, where I had to figure out what I'm going to do when I grow up, basically. When you're a scientist, you do your Ph.D. and you work with mentors who have quite strong ideas of what they want to do. And as a postdoc, you're also part of a lab. But once you have your own laboratory, you can decide to do whatever you like, right? And so that's the time when you have to say, what direction am I going to go into? And so I decided to study genes that increase your risk of type 1 diabetes. And it was a great opportunity.
This is a bit of a tangent, but I'm just curious. Is that a daunting process, or is it exciting? Is it a combination of the two? I mean, it's so much more responsibility than you have at any prior point, working, like you said, kind of helping steer someone else's ship. Rather than just
and I think when you start your own group, some people have a very clear idea of what they want to do. Some people have, maybe a natural transition from their postdoctoral research that would spin out into a larger project that you can start branching into different directions. In my case, I was in a slightly peculiar situation, and maybe we shouldn't spend too much time talking about this because this is a whole podcast in itself. During my postdoc at MIT, I actually discovered that my postdoctoral mentor was fabricating data, and we made allegations of misconduct. There was an investigation, and then he was fired from MIT when I was in his lab. So basically the lab was dismantled. And so it was a slightly peculiar postdoctoral experience, because you're in the middle of a project and your mentor is being fired, and the lab is being taken apart, and everyone's encouraged to move on and find new jobs. So
I think if that had been me, I probably would have gone back to Switzerland to catch butterflies.
Yes.
Like sounds very traumatic.
It certainly was a slightly unusual and stressful time. But and so maybe I didn't get quite the guidance or the opportunity that I could have had if I'd been in a lab where I finish a postdoc and the mentor helps you really pick what direction you're going to go into. So I was left to my own devices a little bit more. And like you say, it's kind of a daunting prospect, because you have to figure out, okay, what are you going to do that's going to be interesting? I mean, there's a lot of interesting questions to ask, but then also something that you'll be able to carry forward for many years and secure funding for. You have to work on something that you'd be able to get other people excited about, so that they actually say, yes, this is a great idea, we'll give him money to do this, which is not always the case, right? So it was a little bit daunting, but then I got some advice from my collaborators who were based in the UK, and at the time they said, "Oh, Stephan, why don't you look at these other genes that we've discovered in these GWAS studies, and that could be good to look at." And so I started to say, okay, this is what my plan is going to be. I'm going to go after these type 1 diabetes risk genes. I'm going to try and figure out their function in disease. And so this is what we did for about 10 years or so. And it was very interesting to do. At the same time, it wasn't quite as rewarding as you might think, because it always takes a very long time to set up these models. So when you make a new mouse model, it's become much faster to make these models because we have better technologies now. But you still, mice still need time to grow up and breed and multiply. And so these projects always take a long time. And then if at the end you find out that the gene didn't really do that much in your mouse model, then you've wasted a lot of time with some rather unexciting results. And so we did this for a number of years, but now we've kind of phasing out this approach of just taking a gene and then making a mouse where you mutate the gene and then find out what it does in disease. Because sometimes we found some very interesting biology and sometimes less so, which is important too, because sometimes you have to say, okay, we thought this gene was important and it turns out maybe it isn't. You've learned something, but it's not quite as exciting as you'd wish for. And so that's where you mentioned earlier these genome-wide screens. And so we did the first one a few years ago. Now, so that was a change of direction for me. So I went from studying one gene at a time, where we know from human genetic studies that the gene's interesting because it associates with diabetes, and we try and figure out the function. And we turned this around where we said, we know the beta cells in the pancreas are being destroyed by the immune system. That's how diabetes happens, right? So these cells that produce insulin are being attacked and destroyed, and then you can't make insulin anymore. So we know that. Is there anything in this beta cell that we can change to protect it? And so the premise is we have no idea what genes might be important, and we'll just blast these cells with constructs to mutate every possible gene, and then we'll see what works. And so, on one hand, we had this for many years. I studied one gene at a time, knowing that it could be important and trying to understand what it does. And suddenly, we just took a completely unbiased approach. We said, we'll just try everything and see if any genes are important, and then we'll figure out how it works later, right? And that turned out to be somewhat successful.
One of the things I'm picking up on is that kind of there have been two major, so the two strategic pushes within your lab have kind of been dictated by technology of the time, in a sense, and changes in our ability to manipulate the genome, right? Or pick out genes from GWAS studies and then look at those genes. And so originally, I suppose, you were looking at things from more of a systems-wide level, but with respect to a single gene. And now the approach is actually, okay, let's change the lens and look at the cell, assume we know nothing, and look at every single gene by using all these constructs which target these different genes.
Right. I think that's exactly right. So, to start with, we're studying one gene at a time with absolutely no idea where the gene was important. So people always assume, it has to be involved in some immune function, but we don't know that. So a gene's important in disease, but you don't even know which cell it's important in, because many cells might express a gene, but for some cells it might be more important than others. And so that's exactly right. So I completely turned it onto its head, if you like, the strategy of saying we know which gene we're interested in, we have no idea where it plays a role, to actually we're going to study one single cell type, and we're going to look at all the genes and see which one's actually important. So it's a completely reversal of strategies, if you like. And you're right that it's dictated in large part by the technology, because this screen we did in beta cells was really facilitated and made possible by CRISPR genome editing, because now we had these libraries, genome-wide libraries, available that you could just purchase that allow you to target every single gene in the genome by CRISPR. I should say, though, that this type of approach would have been possible 15 years ago using RNA interference, because some of your listeners might be aware of the boom in CRISPR genome editing, and there's been a lot of hype around it, and there's been a Nobel Prize just last year. For those of you who are a little older, you may remember that the exact same thing happened with RNA interference. So back in 1997, Andrew Mello and Craig Mello published a landmark paper describing RNA interference in C. elegans, which also earned them a Nobel Prize in 2006. And based on RNA interference, people started doing a lot of studies that now are being done with CRISPR genome editing. But RNA interference is a distant cousin, if you like. It's another way of turning off genes that was used for many years as a prime go-to method before CRISPR came along. And so the CRISPR screen seems to be this new approach, but it isn't, because people used to do RNAi screens back in the day in the mid 2000s, so 2002, 2006, in that area. Especially in cancer biology, a lot of people use RNAi screens to interrogate the genome in cancer cells, for example. The types of screens are not all that different, but I think CRISPR genome editing has made it even several steps easier to manipulate the genome. It's just more efficient than RNA interference. But the same types of large libraries of constructs were made 15 years ago to target the genome with RNA interference, as the libraries that were made just a few years ago to target the genome with CRISPR genome editing.
Accessible, sort of user-friendly, perhaps. Yeah. So I really want to dive into the kind of the weeds, so to speak, of type 1 pathophysiology, because there are really interesting things to consider with respect to, is the beta cell responsible for its own death versus what is the role of the immune system?
The prevailing thought is that your immune system, for whatever reason, starts thinking that beta cells are not right, and that they should be killed. And so the immune cells invade your pancreas, go into the islets of Langerhans, that are these little balls of cells that host the cells that produce insulin and glucagon. And so they go inside there and they start killing off beta cells. And we don't understand yet why they kill off only the insulin-producing cells, but, for example, not the cells that produce glucagon that sit right next to the insulin-producing cell. And looks very much like an insulin. I mean, it's not the same cell; it's a slightly different cell type, but they're very, very closely related. And yet, it's very specifically you only kill off the cells that produce insulin. And so we know it's immune-mediated. We know this from genetics because some of the genes involved are immune genes, particularly the HLA region in the genome that has a very strong effect on the function of T lymphocytes. We also know that if you immune suppress someone, you can kind of slow down or stop the disease. So if you use very strong immunosuppressants, you can stop diabetes from progressing. And so we know that the immune system goes in, kills off these beta cells. And so originally the thought was, okay, there's something wrong with the immune system. For some reason, the immune system thinks a beta cell is dangerous and kills it off. Maybe because it thinks it's like an infected cell or whatever. But now in the last 10 years or so, people have started recognizing that the beta cell wasn't quite right itself. So sometimes, or at least in a number of studies, people have shown that the beta cell might be slightly dysfunctional even before diabetes occurs, and that it may not all be the immune system's fault of mistaking the beta cell for a target, but maybe the beta cell was a target because there was something that wasn't quite right with it. And you can conceive of it. So the immune system is evolved to protect us against pathogens, but particularly against cells that are not quite right. So if a cell is infected with a virus, it'll start waving little flags, and then the immune system knows, aha, there's something wrong with that cell, and it kills off the cell. And at the same time, we'll kill off the virus inside, right? And the same applies, actually, we now know with tumors, so that many tumors may emerge in your body that go completely undetected, at least from us, because the immune system is taking care of them. So the immune system can actually eradicate tumors before they grow. And of course, we still get cancer, and that's because tumors are pretty good at evading the immune system eventually. So you get evolution of the tumor to a point where the immune system isn't able to combat it anymore. but the immune system is very well equipped to kill off cells that are not quite right. And so maybe one of the reasons these beta cells are being targeted is because they're not quite right, and maybe they display signs of disease even though they might not be diseased. And so it could be that both you need a certain dysfunction on the immune side, and at the same time a dysfunction on the beta cell side. And so the balance of this is still...
Which might end up being a sort of positive feedback.
Right. And eventually, the immune attack is going to exacerbate whatever the beta cell has going on. And if you have inflammation in the pancreas, you're obviously going to stress out a lot of cells, including beta cells that are it's not going to do any good. It's probably going to exacerbate the whole autoimmune reaction. But to start with,
Here we can pick up on one detail of... Oh, sorry.
Yeah, go ahead.
I was just going to say, here you can pick up on one particular aspect of a beta cell, which is that basically it's a protein factory. You're producing all of this protein, and that has a stressful effect on the machinery within the cell, which has to pump this stuff out. And so that might also perhaps play... there have been lots of studies on the role of endoplasmic reticular stress, and how that can lead these beta cells to... I'm not explaining this very eloquently, but...
No, but I think... So, the fact is, a beta cell is very specialized in producing insulin, and it makes bucket loads of insulin at any given time, and it secretes a lot of insulin on demand when it's needed. But the insulin is prepackaged because the beta cell has to respond very quickly. So there's a lot of insulin that's kind of prepackaged in vesicles, ready to go, so that when the beta cell sees, a spike in blood sugar, it'll release insulin to bring the sugars down again. And so, like you say, it's a real protein factory. It makes a lot of insulin. At the same time, it's a cell that evolved to do this, so it's pretty well equipped to deal with the stress. So it's quite stressful to produce that much insulin. And I think some recent studies have shown that these cells periodically undergo some stress and then a recovery phase where they produce a lot of insulin and it gets slightly stressed, and then some cells will kind of shut down their production a little bit to recover and then get back online, which is probably also why we probably have more beta cells than we need, and that's sometimes referred to as a spare capacity. So you have a lot of beta cells, but not all of them are working at once. And one explanation might be that you need to have some cells that are able to kind of become a bit more dormant for a while to recover from some stress, whereas others are working full steam ahead to produce the insulin. So the beta cells are in principle equipped to deal with the stress of producing a lot of protein. But then, as you allude to, it might not take that much to bring them out of whack a little bit. So small defects might actually make this stress a little bit too dramatic and then cause some dysfunction in the cell. Or even if the stress becomes too severe, it can even kill the cell. And there are examples of that. So
ER stress can lead to...
Right, so the genetic examples, both from human disease and also in mouse models, that if you just put a little bit of stress on a beta cell, additional stress by misfolding the insulin, for example, it rapidly overcomes the ability of the beta cell to deal with it, and then the beta cells start dying off and you become diabetic. And so there's mutations in the human genome known to cause diabetes solely because of a mutation in insulin folding, and the same in mouse models. So we know that the beta cell can be pushed over the limit of its stress resilience, and that might be something that's happening in type 1 diabetes to a certain extent. So without the beta cell stress necessarily killing the cells itself, but making them just dysfunctional enough that the immune system might be alerted and think, hey, there's something wrong down here. Let's go investigate. Let's start killing off these cells that look like they're not quite right.
So perhaps future treatments of type 1 will involve intervening in this stress pathway as early as possible in disease pathogenesis. So, we spoke about how in the 17th century they were fermenting urine to diagnose type 1 diabetes. Now we can pick up things much earlier on, autoantibodies, different kinds of misfolded insulin. So perhaps, increasingly there'll be pharmaceutical or genetic approaches to preventing that kind of stress from taking off early on. Would that be fair to say?
Absolutely.
And perhaps we could talk a bit about the specific RNLS gene which was found from that CRISPR study we discussed earlier.
Yeah, so let me go back a little bit to what you said. You can maybe predict disease risk in people. So you mentioned autoantibodies. So the best predictor we have are these antibodies you can measure in the blood that recognize antigens derived from the beta cell. And so we now know that typically people measure 5 different antibodies. And we know that if you have two or more of these autoantibodies in the blood, you will develop type 1 diabetes in the next 10 to 20 years. And it could take 5, 10, 15 years, but we know that you will get there. So there are already sure signs that the disease is happening, but it hasn't manifested itself yet with hypoglycemia. And you're not diabetic yet, but we know that you will be. And then even before that, you can look at genetic risk. So we mentioned these GWAS studies, and purely based on genetics, you can predict that someone has a 10-fold increased risk of developing disease, for example, or even absolute risk of 10% of getting disease based on certain genes. And so people have developed these genetic risk scores. And there's some huge efforts on the way, actually, mainly in Europe, where they're actually screening newborns or infants, population-wide, to determine the genetic risk of diabetes. So a lot of these efforts are led by Annette Ziegler in Germany, and they've enrolled many pediatricians who then, just need a drop of blood, and you can measure this genetic risk score in kids. And based on that, you can then enroll people who have an elevated risk in studies where you can then do exactly what you mentioned, right? Prevent the disease very early, even before the stresses occurred in the beta cell, before you have any immune reaction. So that's just as a backstory. So I think at some point it will become feasible to intervene very, very early in disease, rather than, instead of intervening when someone shows up to the clinic and they're diabetic, which is a pretty late point in disease, really. You can envisage how we might be able to treat the disease way, way, way earlier. And then coming back to the beta cell stress, so then if you come in very early, you can maybe even prevent the cells from being stressed out and from causing any kind of immune reaction, right? And there's actually, before we studied beta cells with our CRISPR screen and found this renalase target, other investigators had already done efforts in along those lines, where they looked at genes that were important for beta cell stress and for the death of beta cells in type 1 diabetes. And one of these genes is TXNP. And there's an investigator at University of Alabama, Annette Shalev, who did a lot of work on this. And she found that there's an FDA-approved drug called verapamil that you can actually use to decrease this gene and rescue beta cells and prevent the stress and the stress-induced cell death. And so they, these investigators did an initial small clinical trial, and now there's actually a larger trial being started with this drug to simply reduce beta cell stress and the consequences of beta cell stress to protect the cells. So, these trials are done in patients who were just newly diagnosed with diabetes, with the assumption that these are individuals who still have quite a few beta cells in their pancreas, so that if you can stop the attack completely, you may be able to save what's left of their beta cells and of their insulin and make them insulin dependent. And so our work goes in the same direction. So we did this screen and we found this target renalase, which is a gene that no one really knows the function of. It's an oxidase. It's not exactly clear what it does in the cell. And we also found a drug, FDA-approved drug, that seems to be inhibiting this gene. The drug's called Pargelin. It's an old drug that was used to treat hypertension. It was developed in the 1950s. And at least in our mouse experiments, it shows promise. So, we would love to do trials either with this drug or a related drug to target some of the genes we found in our screen. As you mentioned, to try and protect the cells before any damage has really occurred, before the cells are too stressed. And you could do that even before people have diabetes, so long as you can predict who's at a high risk of developing diabetes.
Interestingly, it's making me think that perhaps, not that this is the wrong question, but the operative question isn't going to be what is the chicken and what is the egg with respect to beta cell and immune system? Because if you can pick somebody up early enough, why don't we just affect both prongs of that? So something we haven't mentioned, but which is undergoing, I think, clinical trials at the moment, is some of the newly formulated CD3 antibodies, which I think have been sort of work on them started about 40 years ago. They had a myriad of problems, but they've been tweaked now that they seem to have an effect if they're given early enough. One can imagine a situation where, okay, somebody has a very high genetic predisposition to type 1 diabetes, and maybe you can detect an autoantibody in the blood. You could give something like perhaps an FDA-approved drug, which will affect ER stress, and also an anti-CD3 antibody, and maybe that will be able to prevent disease progression. So do you think the future will sort of be a two-pronged approach that targets both the immune system and beta cells?
Yeah, I think, until we've found something that works 100% on either the immune system or the beta cell, it's quite likely that we'll have to try combinations. So the mechanisms are very different. You can target the immune system to just stop the immune attack, or you can target the beta cell to make it either more resilient or, not even to alert the immune system to any danger. And I think a combination would work very well. So we know these CD3 antibody studies you mentioned, these clinical trials, are promising. The ones that were done in new onset patients, so patients who were just diagnosed, showed some signs of efficacy, but it really didn't quite stop the disease in its tracks as people hoped for. The latest clinical trials that were done in people who have a very high risk of progressing to disease, so these people I mentioned who have multiple autoantibodies against beta cells, so they will develop disease within the next few years. In these individuals, actually the CD3 antibody treatment was pretty effective in delaying the disease by at least 2 years. And these studies are still ongoing, so we don't know if you may even be able to prevent disease in some of the individuals. We'll know that probably in the next few years. And so right now we don't have any immune therapy that completely stops the disease. And we don't have anything that is going to completely protect the beta cells. But we're making progress on both fronts, and that I absolutely think that combining the two will make sense. Especially, one thing we have to remember in type 1 diabetes, it's a disease that whatever treatment we develop has to be extremely safe, because the alternative is insulin injection. And even though you have to inject insulin multiple times a day for the rest of your life, it's a relatively safe treatment. As far as if you do it right, if the regimen of insulin treatment is correct, it's a safe, sustainable treatment for many, many years. And so you can't have any intervention that is more dangerous than that, really. So you have to develop therapies. If you think cancer, cancer treatments, for example, cancer treatments can be very invasive, have severe side effects, because they're going to save your life. That's an imminent danger from cancer, sometimes within the next few months or a couple of years. That's not the case for diabetes. If you treat someone with insulin who has diabetes, you can't use anything that has severe side effects or puts their life in danger. So we have to be somewhat conservative with the interventions we use. And so that's maybe why they're not as efficacious as they could be, right? So
especially because the relative risk of now, people have closed glucose pumps which function actually very well, right? So the trade-off becomes increasingly like the margins thinner and thinner, I suppose, in terms of like acceptable risk. Yeah. I'm aware we're sort of rapidly running out of time, even though I feel like we could get into a lot more molecular sort of detail. But yeah, in the interest of time, sort of looking forwards, what approaches, sort of what new projects, what new ideas have got you really excited, and perhaps members of your lab really excited?
So really, I think that this latest effort we've had in trying to identify genes we can modify in beta cells has been really exciting for us on two fronts. First, and as you know, because you work in a laboratory that Doug Melton's laboratory that is kind of one of the leaders in the field of stem cell beta cells, we can now make beta cells from stem cells, and we can make stem cells from people's blood samples. So we can take someone's blood, turn it into induced pluripotent stem cells, turn these stem cells into beta cells, and put them back into the patient. So in principle, we know how to make beta cells to cure a patient. We just have to figure out what do we need to do to these cells to protect them against autoimmunity. And so the project we did in the lab with my colleague Peng Yi at Joslin, we found genes that perhaps could protect these cells if we mutate them. And so we're very excited to test these ideas so that we could take stem cell beta cells, mutate certain genes that we think are going to be protective, and then put the cells back in a patient and possibly cure diabetes, right? So that's the prospect. It's not going to happen overnight. We're not there yet, but just the idea that we can do this and that we already have a proof of concept, if you like, from our mouse studies is very exciting. And the other thing is that we found some of these genes that are protective in beta cells that could actually be drug targets. And so that would actually allow you to protect the cells without modifying their genome. And even without putting new cells in, you could find a drug that can protect a beta cell. You could actually give the drug before the beta cells are killed in someone who's at risk of developing diabetes. Just like the trial I mentioned with verapamil, that's a drug that's protective for beta cells, or at least to a certain extent. The idea is you could give that early on to prevent the beta cells from being killed in the first place before someone's diagnosed. And the same with the drugs we're trying to develop to target the renalase gene we discovered to be protective. So I think there's some very exciting prospects on two fronts. One is to modify the genome of stem cell beta cells so that you can put protected cells back in a patient and cure diabetes. And the second is to develop drugs that specifically protect beta cells in someone who doesn't have diabetes yet, but is going to. So someone who has multiple autoantibodies who we know will develop diabetes in the next 5 or 10 years, you can give them a drug. If it's a drug that's cheap and safe and easy to take, you might be able to completely prevent the disease from progressing. So that's what I think is very exciting right now.
Yeah, absolutely. Interestingly, sort of both of those approaches fall on opposite ends of the kind of cost spectrum, right? Using FDA-approved drugs versus trying to make beta cells from each patient and then get those cells to, sort of through regulatory approval for good manufacturing practice, et cetera, especially if you've done genome modification to those cells. I'm asking you for a snap judgment here, but how realistic do you think is the cost barrier to using an autologous cell, so a cell from a patient to cure their own disease, which you've reprogrammed, is that barrier going to be surmountable? And if not, do you think that an allogenic cell, so a cell from embryonic cells, do you think that has serious potential?
So the question really is, are we going to have to rely on an off-the-shelf cell product, right? That a company makes a gazillion cells of one type and it gives it to every possible patient, or whether we can have kind of a bespoke, tailored solution where every patient gets their own cells made for them. And clearly, off the shelf is always cheaper because you make one product and you can validate it and test it, and then you just make a bunch of it. But the ideal solution might actually, I think in the long run, the ideal solution would be to have kind of a bespoke, made-to-measure precision medicine type approach where you take a patient's own cells, make them into stem cell beta cells, and put them back in. And right now, it's prohibitively expensive to do this for individual patients. We would think that's not a viable option. But as we develop these methods to make stem cells, as we develop and scale up methods to make stem cell beta cells and optimize protocols, the manufacturing, the testing, everything can be streamlined. I would foresee that in 5 years, 10 years, it might not be an issue anymore. So the cost might not be an issue. If we're able to routinely make stem cells and stem cell beta cells from any given person at a reasonable cost, I think that would be the way to go. And that's more of an engineering problem, it's more of an engineering problem than a biological problem, because we know we can do it. It's just a matter of how can we do it more cheaply. So it's not something.
It's sort of like the cost of like the cost of genome sequencing, or
right, exactly. Yes, the ideal cure for someone who has type 1 diabetes is to give them back their own cells that are perhaps engineered to be protected, or just give them back their cells and give them a drug to go with it, or an immune treatment. But I think getting your own cells back would be the best way to go.
Do you think that the genome modifications? Sorry, last question. I know you're sort of time-wise we're kind of out of time, but I can't help but ask, do you think the gene modifications to those IPS lines, sort of similar to how we were talking about prevention, will involve targeting genes involved in perhaps responses to ER stress, as well as perhaps genes involved in immune recognition? So do you think that expression of things like PD-L1 and CTLA4, these are sort of signals to T cells that we've sort of spoken about, well, T cells, not the signals, but will be a useful combination with something like...
Well, my take on it is that since we still don't understand why a beta cell is targeted in the first place, there may be a reason why the beta cells become target of autoimmunity that has nothing to do with expression of immune genes or anything. There might be something fundamentally off with these cells that we might be able to fix that doesn't involve adding in lots of immune suppressive genes and hiding the cell from the immune system. And that's kind of, in a way, the agnostic approach we've taken in our Crispr screen that we published, where we said we have no idea what's going to work. And then we found genes that, are completely random at first glance. You look at these genes, and I'd never heard of any of them, and they have nothing to do with immune function or immune recognition. And it could well be that you can modify a beta cell in its function to make it look perfectly healthy to the immune system, and the immune system just leaves it alone, regardless of what MHCs you express and whatever. So that's a possibility I'm open to, and it's something that we're investigating right now. The idea that you can just maybe fix something quite fundamental in a beta cell to make it just look fine, look healthy. The immune system just, is told, like, move along, nothing to see here, no reason to attack me.
I suppose the presupposition there is that pathogenesis in all type 1 diabetics essentially follows the same sequence of steps. So it might be that people with a different set of genes, risk genes, it's more immune mediated to begin with and actually has less to do with the beta cell. I'm making this up as I go, but yeah, okay.
Well, I think you're right. I mean, it might be quite heterogeneous. So, some people might have a more aggressive immune system. Some people might have a beta cell that's more dysfunctional, and it's a mixture of these two that ultimately, brings you to the point where you get type 1 diabetes. Some people have a very autoreactive immune system, and just a little bit of a defected beta cell might cause disease. And in other people, the immune system is slightly off, and then the beta cell is quite defective, and that brings you to disease. So the balance of the two might be very different. And so you're right that fixing the beta cell might work very well for people who have mostly problems with the beta cells, but maybe not so much for people who have a beta cell that's almost normal and where the immune system is particularly aggressive. So it's a good point. We have this heterogeneity. So, and that brings us back to the point where you might have to combine an intervention that targets beta cells with an intervention that targets the immune system, and maybe on a case-by-case basis, at some point we'd be able to evaluate who benefits most from one or the other, or do you need both?
Yes, perhaps you could combine, like, sort of sequence everyone who's subject to T1D, and then down the line when you're using different therapies, see what works best for different classes of patients, essentially. Fantastic. What a brilliant place to end on. I'm aware we're out of time, but yeah, Stephan, thank you for joining me. I had just really enjoyed hearing, a bit of a history lesson and hearing about your works.
Well, thank you. It's a pleasure to talk to you.
Thank you for listening to the Pager podcast. If you've enjoyed it, then head to Spotify or iTunes to hear more.
As ever, we'd love to hear what you think, and you can reach us via our Facebook page or by email. See you next time.